Hey champs, I'm actually on a short trip. Oops, I forgot to welcome you to 90 Plus My Tuition app. I hope you all are safe and strong. You know what, I'm quite excited today because I am going for a jungle safari and also because we are going to deal with chapter number 5, the fundamental unit of life, where we will be dealing with what living organisms are made up of. Well, aren't you excited to know the reason for my excitement? I hope so. Oops, my stop reached. We will meet soon. Bye-bye. You might be wondering that I am here for these tigers, right? Well, that's not the actual case. I am here for this particular honeycomb. And you might be wondering why, right? Well, it's because this honeycomb reminds me of a story that I learned a long time ago. It's about the famous scientist Sir Robert Hooke, who was in a quest to find something really interesting. He was working on thin slices of cork. Well, remember that this is an old story because all this happened in the year 1665, which seems pretty long back, right? And for those of you wondering what a cork is, let me tell you, it's the part of the bark of a tree. And you know what? It helps the tree by preventing excess water loss. By the way, we were talking about Sir Robert Hooke, right? So let's come back to the famous experiment that he did. You know what? He used a microscope that was self-made because there were no developed microscopes back then. And with this simple microscope, he could only observe few box-like structures which had walls for separation. Well, if you notice, you will find that this structure has a high resemblance to that of a honeycomb. Right? Yes. So, if you look carefully, you will notice few box-like structures here, right? And one fine day, he decided to name this particular box-like structure as a cell, which means a little room in Latin. It seems like one, right? And now comes a big twist. The cells that he observed were dead. And for viewing and understanding living cells, he needed improved microscopes, which, as you know, weren't available back then. I know that's sad. But don't be sad, because his accomplishment made him the discoverer of cells, which is a piece of great news, right? Well, as slowly time went on, other scientists also came up to the limelight with the invention of magnifying lenses and newer technologies that led to the discovery of the microscopic world. And our next hero, Anton van Leeuwenhoek, was one among them. In 1674, he found a unicellular, free-living algae found usually within ponds and lakes. Welcome to Kaun Banega Arabpati, and the very first question for today's show is, who observed a living cell for the very first time? And your options are option A, Robert Hooke, option B, Anton van Leeuwenhoek, option C, Robert Brown, and option D, Jan Evangelista Perkin J. And your time starts now. Well, if your first thought was about Robert Hooke, then it's absolutely fine, because almost everyone thinks like that. But the correct option is option B, Anton van Leeuwenhoek, because he was the one who observed a living cell, that is a unicellular free-living algae, for the very first time. And on the other hand, Robert Hooke was the one who observed a cell, that is a cork cell, but this cork cell was actually dead. So with this, I conclude the show. Have a great day. We are talking a lot about cells, right? Don't you wish to take a look at it? Well, this is a cell, and if you look closely, you will find that within the cell there is an important organelle known as the nucleus, which was found by another great scientist, Sir Robert Brown, in the year 1831. Well, it didn't stop there. In 1839, Jan Evangelista Purkinje, or let's call him Sir Purkinje, found a fluid substance within the cell. He then decided to coin a new term for it. He called it protoplasm. And you know what? This protoplasm is actually made up of two different regions, that is the cytoplasm and the nucleoplasm. Well, if you don't know what these regions are, I'll tell you. The region that you see within the cell is known as cytoplasm, and the region present within the nucleus of the cell is known as the nucleoplasm. So now you know what protoplasm is, and you also know that the protoplasm is made up of cytoplasm and nucleoplasm. Okay, champs, so now we know that who all discovered a cell, a living cell, the nucleus and the protoplasm, right? So now I guess it's time to stop discussing about these things, because we urgently have to meet two great scientists who introduced cell theory. Well, it's none other than Schleiden and Schwann. They contributed to the cell theory in different years. Schleiden contributed in the year 1838 and Schwann in the year 1839. And together they put down a theory which stated that all the plants and animals are made up of cells, and that the cell is the basic unit of life. Well, it seems like Sir Virchow was interested in expanding the cell theory. In 1855, he suggested that all cells arise from the pre-existing cells, or in simple words we can say that if this is a cell, then according to his theory, it might have arisen from the division of another old cell. I hope you understood this. Okay, fine. I agree that I showed you a picture of a cell, right? But this time I want to show you a real cell. Well, for that we need to do an experiment. So come, let's do it inside a laboratory. Okay, so here we are. First of all, you need to take a piece of the fleshy leaves of the onion bulb, and with the help of a pair of forceps, try to peel off the epidermis from the inner surface or the concave surface of this peel, and then immediately transfer it into a watch glass containing water to avoid folding and drying of the peel. Further, take a glass slide and put a drop of water in the center. Well, you know what? You will need a paintbrush to transfer the peel from the watch glass to the glass slide. Well, make sure that the peel is flat and not folded. Okay? And now we will need a drop of saffron in solution for staining the peel, so that the cells of the onion could be visible under the microscope. Well, after that, you need to put the cover slip at a 45-degree angle over this glass slide before viewing it under the microscope. Well, make sure that you do this step really carefully for avoiding the formation of air bubbles. And now finally, you can put this glass slide under the microscope and view it. Well, make sure that you first view it under lower magnification power and then later under higher magnification power. Okay? Let me peep through this. Oh, I see so many small structures arranged together here. Well, meet them. They are cells. They are the basic unit of all living organisms. Well, now if I ask you this question, that is, what are living organisms made up of? You might already know the answer, right? Yes, living organisms are made up of cells. And remember that this is how the onion cells look under the microscope. Interesting, right? Oh, I have a doubt. What if I take a bigger onion, and a small onion. What do you think? Does the bigger onion contain bigger cells? Well, if that's what you think, then it's absolutely wrong. Because the size of the cells will be the same, but the number of cells will be more in the bigger onion, which makes it bigger in size. Okay? Actually, number of cells is an interesting topic. Do you know that there are organisms who are made up of just a single cell? Well, yes, organisms like amoeba, chlamydomonas, paramecium and bacteria are made up of just a single cell. And that's why they are known as unicellular organisms. Well, yes, their whole body is just a single cell. Oof, I wonder how they manage everything. Well, now on the other hand, there are organisms like animals, plants and fungi which are multicellular organisms. They arise from just a single cell, that is a zygote or a fertilized egg, which undergoes rapid cell division to form cells of different sizes and shapes. And finally, it gives rise to a whole new organism, which sounds amazing, right? Oh, did I say shape? Well, do you know that this amoeba has no particular shape? Well, yes, it's an irregularly shaped organism. Imagine if we were like that. We could literally seep into a room. But no, most of the cells of our body have a fixed shape, like that of this neuron. Hey, wait! It seems like these WBCs or white blood cells, which are responsible for protection against harmful pathogens, are not in this group of fixed shape cells. Well, yes, they are not in this group. They are actually irregularly shaped, just like that of an amoeba. Well, since we are talking about the shape of cells, we will do a fun activity. I will ask you a few questions after showing you the pictures of the cells that I saw when I observed the temporary mounds of the onion peel, the onion root tip and the leaf peel under the microscope. Well, look at them really carefully and then try to answer my questions. Fine. And here comes my first question, that is, do you think that the cells look alike in terms of shape and size? Well, look at this picture and tell me, do they look alike? No, right? They don't look alike. That is, all the different types of cells have different shapes and sizes. And now comes the second question. Do the cells look alike in terms of their structure? Well, externally they might seem similar, but internally they are not same in their structure. Fine. Time for the third question. Could we find differences among the cells belonging to different parts of the same plant body? Well, yes, definitely we can find differences because, as you know, different parts of the plant body are associated with different functions, right? For example, just think about the leaf cells. They are associated with photosynthesis. So obviously, they will have cell organelles such as chloroplast which are associated with this function in a much more amount than any other plant part. Well, this was just one single example. There are many such differences about which you will learn in your higher classes. And now comes the last question. What similarities could you observe between these cells? Well, there are certain similarities. That is, certain structures inside the plant cells are common to all, like the cell membrane, cell wall, and the cytoplasm. Well, usually all the plant cells also contain the nucleus. Oops, I forgot to share a memory. You know what? Once I got to see what's inside a cell. I was quite amazed to see all this. And that's when I got the answer to most of my questions. That is, what a cell is made up of and what is the structural organization of a cell. I got to see that a cell is made up of different organelles. And you know what? These organelles have divided their chores or function. And this is known as division of labor. Well, here I saw that few organelles were making proteins. They look cute, right? And few others were degrading and digesting waste. Seems like a tough job, no? I also noted one thing, that is, despite their different functions, almost all different types of cells have the same set of cell organelles. Well, there are exceptions too, like our RBCs or red blood cells. You know what? They do not have a nucleus and a mitochondria. Well, these cell organelles are new to you, right? But you will be studying about them really soon. And now, if someone asks you why the cell is known as the structural or the functional unit of life, you already know the answer, right? The cell is the basic unit of all living organism or the building block of life, which divides rapidly, giving rise to a whole new organism. And at last it gives it a structure, which makes it the structural unit of life. Ta-da! And now, as the cell performs different functions, may it be metabolic or physiological, it becomes the functional unit of life. Well, tighten your seat belts because you will get to know the different functions of a cell really soon. But as of now, I guess it's time to meet the security guard of the cell, that is the plasma membrane. As you can see, it's the outermost covering of the cell. And you know what? It is flexible. And it is made up of organic molecules like lipids and proteins. Well, it actually separates the content within the cell from the external environment. And it also manages the transport of materials from within the cell to outside. I know that's interesting. Well, as we have seen and we are talking about the plasma membrane, I have a quick question for you. What if the plasma membrane ruptures or breaks down? What will happen? I know that this is a very interesting question. Well, if the plasma membrane or the cell membrane ruptures or breaks down, then the regulated exchange of the contents within the cell and the cell surrounding won't be taking place. And moreover, the contents within the cell will actually get exposed to its surroundings, right? Hence, there will be a loss of the protoplasm, which will eventually lead to cell death. Well, that's a very tragic end, right? You know what? I need to tell you something else too. But for that, we should move out of the cell. So, come on. And hey, do you know that for viewing the structure of the plasma membrane, we will be needing an electron microscope? Yes. And before going any further, let's see the picture of the electron microscope. It seems really huge, right? It's huge and it's an amazing tool to observe the ultrastructure of the cell because of its high resolving power. Because of this particular feature, it produces high resolution images. And as the name suggests, in the case of electron microscopes, electron is used to illuminate the object. Well, here, these electron beams are generated by heating the tungsten filament. You might be thinking how the tungsten filament is heated. Well, it is with the help of high voltage current that the tungsten filament is heated, and that's how an electron beam is produced. Interesting, right? And this is how we get to see the plasma membrane. And now let's continue talking about the plasma membrane. You can think of it as the security guard who allows the entry of few materials within the cell. And on the other hand, at times it do not allow the entry or exit of some other specific materials. And that's why it is known as selectively permeable membrane. So now if someone asks you, that is, why the plasma membrane is known as a selectively permeable membrane, you already know the answer, right? And since we know the answer, let's move out of here, okay? Hey, how do you think this movement occurs? Well, before we start anything, remember this code. Gaseous diffusion and liquid osmosis. Here, diffusion is the spontaneous movement of substance from a region of higher concentration to a region of lower concentration, and it is important for the exchange of gases and water in the case of cells. Well, apart from this, the nutrients are also transported with the help of diffusion. And if you still didn't understand the definition of diffusion, then no worries, because we will learn this with the help of an example, okay? Think of the gas carbon dioxide. It's actually a cellular waste, and as it's a waste, it needs to be removed out of the cell, or it might get accumulated within the cell and cause trouble, which is something that we really do not want. And here comes the role of diffusion. The carbon dioxide which is present in a higher concentration within the cell diffuses out to a region of lower concentration, that is, towards the cell's surroundings. So now we saw the very first example of diffusion. And now something different happens in the case of O2 or oxygen gas. Here, due to rapid consumption of oxygen within the cell, the concentration of oxygen within the cell will be quite low in comparison to its surroundings. Hence, diffusion will occur, of course, but this time it will occur in the opposite direction. That is, oxygen will move from a region of higher concentration, that is from outside the cell, to a region of lower concentration, that is within the cell. Did you see the difference? Did you notice that both carbon dioxide and oxygen are gases and they move in and out of the cell with the help of diffusion? Well, that's why I told you to remember the code gaseous diffusion, because gases usually moves in and out of the cell with the help of diffusion. Okay? Now let's talk about the code liquid osmosis. Here in the case of osmosis, the movement of solvent molecules occur from a region of higher concentration to a region of lower concentration through a selectively permeable membrane. Here note that the solvent molecules are usually water molecules, okay? And do you know what happens at the end? Well, yes, equilibrium is reached, which means that the concentration of water will be equal on both the sides. That's interesting, right? You will understand this with the help of just 3 different situations. But before that, remember that the solution in which we dissolve something is known as the solvent and the dissolved substance is known as the solute. For example, if we take a glass of water and put some salt into it, here the water is the solvent and the dissolved substance, that is the salt, is the solute. Okay champs, it's time for some fun. Here imagine the very first scenario where the sugar or the salt containing medium contains a cell within it. Okay, we have put the cell right now. And now also remember that this sugar or salt containing medium has a high amount of water. Or we can say that it is highly diluted or it has high concentration of water in comparison to the cell that we placed inside the solution. Okay, now think what will happen. In which direction will the movement of water occur? Come on, think. Well, of course, with the help of osmosis, the water molecules will be moving in both the directions. But more water will be moving from a region of higher concentration, that is from the salt or sugar solution, to a region of lower concentration, that is within the cell. Hey, did you notice that the water molecules are moving within the cell? What will happen now? Obviously, the cell will eventually swell up. That's very interesting, right? Oh yes, so this type of solution which has a higher concentration of solvent and a lower concentration of solute is known as a hypotonic solution. And when the cell is put inside a hypotonic solution, it swells up. Okay? And now comes the second scenario where the concentration of water in the surrounding is equal to the concentration of water within the cell. Now tell me, in which direction will the water move more? Well, to your surprise, the water molecules will be moving in both the directions. But the amount of water that is going in and moving out will be equal, which makes the net flow of water molecules a big zero. And this type of solution is known as isotonic solution. And now comes the third scenario where the outer medium contains literally low concentration of water in comparison to the cell placed within it. And now don't you think that this time the water molecules will move out from the cell towards the outer media, as this time is the cell that contains higher concentration of water in comparison to its surroundings? Well, that's absolutely true. And as the water will move out from the cell to its surrounding media, the cell will shrink. I know that's sad. And you know what? This type of solution which has a lower concentration of solvent and higher concentration of solute is known as hypertonic solution. And when a cell is placed inside a hypertonic solution, it shrinks. Hey, you know what? This osmosis is actually a special case of diffusion which occurs through a selectively permeable membrane. Remember that, it's very important. And now it is actually seen almost everywhere. For example, if we take a look at this plant, it takes in water through its root with the help of osmosis. And the same happens in the case of unicellular freshwater organisms. They also take in water with the help of osmosis. Champs, do you want to see osmosis in real life? If yes, then it's experiment time. Come, let's move to the lab. Okay, so here first of all, you need to take an egg and treat it with hydrochloric acid or HCl. Well, this will help in creating pores in the eggshell, which is made up of calcium carbonate. And now guess what will happen? Well, here now just a thin outer skin will be there to enclose the egg. And this thin outer skin is actually permeable. Now we need to put this egg into a beaker containing pure water and wait for 5 minutes. Let's see what happens. Can you guess? Okay, I'll tell you. The egg actually gets swollen because the water molecules from the pure water will move inside the egg or the cell. Egg is a cell, right? You know, I suppose. Well, this happens because of osmosis. Well, what will happen if I place the deshelled egg which was inside the HCl solution into a beaker containing concentrated salt solution where the amount of water molecules is really very less? Guess what will happen? Fine, I'll tell you. The egg or the cell will actually shrink because the water molecules will move from a region of higher concentration, that is from within the cell, to a region of lower concentration, that is to its surroundings. Oh, that's amazing, no? Hey, time for a quick question. What will happen if I put dried raisins or apricots inside a beaker containing plain water? Guess? Okay, I suppose that you guessed it right. Yes, the dried raisins or apricots will actually swell up because the water molecules will be moving from a region of higher concentration, that is from the plain water, towards inside the dried apricots or raisins where the water concentration is quite low. I know that this was interesting, and it is actually a true example of osmosis that we can see in our everyday life. Okay, so now we know that how substances like CO2 and water move in and out of the cell, right? Well, yes, it's because of the permeability of the plasma membrane, which allows the exchange of gases through diffusion and water molecules through osmosis only when there is a concentration difference across the cell. So, all thanks to the permeability of the plasma membrane. And now the very next talent of the plasma membrane is its flexibility, which actually allows the cell to move and also to take in food and other necessary and very important things from the surroundings. Well, this could be easily observed in the unicellular amoeba. Here, Mr. Amoeba engulfs its food with the help of pseudopodia, which are actually temporary structures that appears and disappears like magic, and they are usually used for locomotion. But pseudopodia also help in creating food vacuoles, which stores the food. And you know what? The amoeba later releases certain digestive enzymes which breaks down this food. Well, this whole process is known as endocytosis. So that's all about the flexibility of the plasma membrane. Hey, do you know a fun fact? A cell membrane or the plasma membrane is considered important for a living cell as it controls what comes in and what goes out and does a lot of other functions too. And since viruses do not have a cell membrane, they are not considered to be living until and unless they infect other living organisms and use their machinery. This is something really interesting, right? Okay, I guess it's time to start something new, right? Do you know that plant cells, fungi and bacteria have another extra outer covering apart from the plasma membrane? Well, oh yes, it contains, and this layer is known as the cell wall. Well, it is present just outside the plasma membrane, and in the case of plants, this layer is kind of rigid because it is composed of a component known as cellulose. And this is the reason why plants have additional strength. And you know what? I noticed that when I put a plant cell inside a beaker containing a solution having lower water concentration, the plant cell immediately lost water. And this also caused the movement of the cell's content away from its cell wall. Well, I was shocked to see this. And this process where we can see shrinkage or contraction of the contents of the cell away from its cell wall has a name. It's known as Plasmolysis. Now, on the other hand, if I take a plant cell, a bacterial cell or a fungi and put it inside a beaker containing a very dilute medium, or let's say a hypotonic medium, or a medium that contains higher water concentration, then guess what will happen? Come on, guess. I'll give you 5 seconds time. 1, 2, 3, 4, 5. Okay, I'll tell you. The cell actually swells up. And here I noticed one thing. That is, the cell wall acted as a rigid outer covering and prevented the cell from bursting when excess water tried to get inside the cell. I know that the cell wall did a commendable job. And that's why I'm a big fan of the cell wall. Well, you know what? The cell wall actually helps the cells like the plant cell or the bacterial cell and the fungi to withstand greater changes in the surrounding medium than the animal cell. That's something really interesting, right? Well, we learned about plasmolysis, right? Well, now we will see an experiment which will make you understand things in a better manner. Okay, so for this, you will need a real plant. And now we will take a leaf peel and mount it on a glass slide containing water and then view it under higher power of a microscope. Oh, I see so many green granules present here. Well, these green granules are known as chloroplast. And do you know why they are green? Fine, I'll tell you. They are green because they contain a green pigment known as chlorophyll that helps in the process of photosynthesis. Oops, that's not the topic here, right? Fine, let's come back. Okay, now we will put a drop of strong sugar or salt solution over this mounted peel and view it again under the microscope and see what will happen. And what do we get to see? Well, here we can see a cell that has gone shrunken. Well, it's because the cell's content actually moved away from its cell wall. Why did all this happen? I will tell you. It's because the water moved from a region of higher concentration, that is from within the cell, to the surrounding medium where the water concentration was really low. Because the surrounding medium contained strong solution of sugar and salt, right? So here we actually saw plasmolysis due to osmosis. Fine, now I suppose we'll add some more masala into this experiment. We will take a ryu leaf and put it inside boiling water and wait for a few minutes. And then we will transfer it onto a glass slide and then view it under a microscope. Oh, here I can see cells with damaged organelles. Or I can say that these cells are actually dead. Fine. Now we will repeat the same procedure that we did before with these dead cells. We will treat them with strong sugar or salt solution and see what will happen. Are you guys excited? Because I am so much excited. Well, to your surprise, plasmolysis won't be taking place here because the cells are dead, right? And here the plasma membrane, which allowed the exit and entry of water molecules within the cell itself, is damaged. Then how will this process occur? So with this we can conclude that only living cells show osmosis, and this is how we can see plasmolysis too. Okay, now I guess it's high time to talk about cell organelles which are present inside the cell and which are the ones that manage everything that happens within the cell. Okay, so the very first guy or the cell organelle is the nucleus, which is actually something that fascinates me always because it does a really great job, and that's why I named it as head of the department or the cell. Well, I saw a clear nucleus for the very first time when I did an experiment with the cheek cells. For this, I had to scrape the inside surface of my cheeks gently and then place or spread the material over a glass slide. Okay, and then I needed to put methylene blue over this and view it under the microscope. And do you know what did I see? Well, I'll tell you. I saw a dark colored spherical or oval dot-like structure near the center of irregularly shaped or roughly circular shaped cell, and this structure was the nucleus. And now if we take a proper look at this nucleus, we will find that it has a double membranous covering. Well, it is known as nuclear membrane. And you know what? This nuclear membrane have pores so that the transfer of materials could happen between the nucleus and the cytoplasm. That is both the ways, inside to outside and also outside to inside. Well, I already told you that cytoplasm is found around the nucleus, right? When we learned about the protoplasm, remember? Oh, and you know what? There are few organisms like this bacteria who do not have a nuclear membrane, which is why we can say that they have poorly defined nuclear region. But you know what? They have an undefined region that contains all the contents of the nucleus. Well, this region has a name. It is known as nucleoid. And these type of organisms who do not have a nuclear membrane are known as prokaryotes. Now, on the other hand, there are organisms like us who do have nuclear membrane that envelopes our nucleus, and that is why we human beings are known as eukaryotes. Well, there are a lot of differences in between prokaryotes and eukaryotes, which we will be learning soon. The eukaryotic cell have a size of around 5 to 100 micrometers. Now, on the other hand, prokaryotes are generally smaller in size. Their size is around 1 to 10 micrometers. And you know what? They usually lack few membrane-bound cell organelles, or we can say that they lack organelles which are enveloped by membranes. For example, mitochondria and the chloroplast. Well, so the functions of these particular organelles are done by poorly defined regions of the cytoplasm in the case of prokaryotes. Now, on the other hand, eukaryotes have these membrane-bound cell organelles. Let's talk about an organelle here for making things more understandable, right? So, I guess we should go with chloroplast. And I know that we learned about chloroplast a few minutes back. But then here we need to compare between a eukaryotic cell and a prokaryotic cell, right? Here the eukaryotic cell that contains the chloroplast is the plant cell. And you know what? This chloroplast is actually a membrane-bound plastid which contains green pigments known as chlorophyll. And these chlorophyll molecules are responsible for photosynthesis, and they are the ones which actually causes the coloration of the leaf. That is, it gives a green tinge to the leaves. I know that's interesting. And now we will see about prokaryotes. But in the case of some photosynthetic bacteria which are prokaryotes, this is not the case. The chlorophyll pigments are not present within the plastids where it is usually seen. Instead, they are associated with membranous vesicles. Now do you see the difference? Yes, this was the thing that I was talking about. And as we have finished this, let's move on to the nucleus again. So, apart from the nuclear membrane, the nucleus contains another very important thing. Can you guess? Fine, I'll tell you. Have you heard about chromosomes? Well, they are the ones which carries information regarding all the characters from the parents to the offsprings in the form of DNA or deoxyribonucleic acid. Well, apart from the DNA, the chromosomes also contain certain proteins. Okay, now I'll tell you a fun thing. That is, the functional segment or the actual functional segment within the DNA have a special name. It is known as genes, and they are the ones which are responsible for carrying the information from the parents to the offsprings. So now we have learned about chromosomes, about their proteins, the DNA or deoxyribonucleic acid, and also about genes. Okay? Hey, wait for a second. Look at this picture. It's a chromosome, right? But no, it doesn't always look like this. It is actually visible as an entangled mass of thread-like structures known as chromatin materials. And you know what? When the cell is ready to divide, these chromatin materials get organized into rod-shaped chromosomes like this. I know that it is amazing, right? Well, the HOD of the cell or the nucleus does more amazing things like controlling cellular reproduction, which is a process where the cell divides into two daughter cells. And after the reproduction, it also determines the way in which the cells will develop and also what form it will exhibit at its maturity. Okay, it does all these jobs by controlling the chemical activities within the cell. Seems like a pretty tough job, right? Hey, have you ever wondered where all the cell organelles float? I'll tell you. If you look inside the cell membrane, you will find a fluid substance or a fluid region. Well, this is where all the cell organelles are found. And you might be quite familiar with this region. It's none other than the cytoplasm. And if you look closely at the stained cheek cells and the onion cells, you will find that cytoplasm is the region which takes up just a little stain in comparison to other regions. Seems interesting, no? Since we are talking about the cytoplasm, I will ask you a quick question. Why do you think that the cell organelles present within the cytoplasm are membrane bound? Think about it. Any guesses? Fine, I'll tell you. Champs, I already told that the cell organelles show division of labor, which means they do different functions or that they have different job roles to perform within the cell, right? So to keep these different functions separate from each other, they are membrane bound. I hope that you understood this. Now look at this lysosome sweeping out the waste and the Golgi bodies parceling the proteins. They both do different functions. And for doing different functions, they should be separated from each other, right? So here comes the role of the membranes. Oh, I guess that we should seriously learn some management skills from them. Am I right? Champs, do you see this structure? It's none other than ER or Endoplasmic Reticulum. And you know what? It is actually membrane-bound tubes and sheets. And if you look closely, it looks like long tubules or round or oblong bags or vesicles. And it has a similar structure to that of a plasma membrane. I know that you don't feel the same. Even I don't feel the same. But it's the actual truth. And you know what? This ER or Endoplasmic Reticulum are of two different types. Rough Endoplasmic Reticulum or RER and Smooth Endoplasmic Reticulum or SER. Do you know why RER or Rough Endoplasmic Reticulum is known as Rough Endoplasmic Reticulum? Any guesses? Fine, I'll tell you. It's because they contain structures known as ribosomes on their surface, which actually gives a rough look to the endoplasmic reticulum when viewed under the microscope. And you might be wondering, what role does this ribosomes perform, right? Well, they are responsible for protein manufacturing. And after the proteins are made, the ER or the endoplasmic reticulum serves as a channel for transporting the proteins throughout the different regions of the cytoplasm or between the cytoplasm and the nucleus. Seems like a big task, no? Now, on the other hand, if we talk about smooth endoplasmic reticulum, it helps in the synthesis of fat molecules and also lipid molecules. Well, you know what? There is something else. That is, in the liver cells of animals coming under phylum Vertebrata, this SER or smooth endoplasmic reticulum also helps in detoxifying poisons and drugs. That's something really interesting, right? So now you know that where does the lipids and fats necessary for cell membrane synthesis occur, right? I hope you are amazed. Well, yes. So now you know that the ER or the endoplasmic reticulum plays a major role in the synthesis of proteins, fats and lipids, right? And all this is used in the synthesis of the cell membrane. Well, you know what? This process has a name. It's known as membrane biogenesis. Well, apart from this, the proteins and the lipids are also used for the formation or the synthesis of hormones and enzymes. Well, that's interesting, right? And there's one more very important thing. The ER functions as a framework inside the cytoplasm of the cell and provides a surface for many biochemical activities to take place within the cell. That's much more interesting. Look at him. Do you know him? Well, he is Camillo Golgi. Say hi. He is the one who discovered the Golgi Apparatus. and the Golgi apparatus is actually membrane-bound flattened sac-like structure or vesicle that are arranged parallel to each other in stacks called cisterns. And you know what? The membranes of this Golgi apparatus are in close connection with the membranes of the ER or the endoplasmic reticulum. So what all materials are being synthesized near the ER or the endoplasmic reticulum are packaged and transported to different regions within the cell and also towards the outer surface of the cell with the help of this Golgi apparatus. I know that it does a commendable job. You know what? You should think of the Golgi apparatus as a post office because here storage, modification and packaging of products within the vesicle takes place, and it actually helps in the transportation too. Well, apart from this, here the complex sugars are formed from the simple sugars. That's something really interesting, right? And there is one more really important point. That is, here the formation of lysosomes, which is another cell organelle, also takes place. So now you know or you can guess that what would happen to the cell if there was no Golgi apparatus. It would have been a disaster. And hey, I mentioned lysosomes, right? Do you know that they are known as suicide bags? Interesting, right? Well, do you want to know why they are known as suicide bags? I'll tell you. It's because these lysosomes are actually membrane-bound sacs which are filled with digestive enzymes. And remember that these digestive enzymes are synthesized within the RER or the rough endoplasmic reticulum. Okay, do not forget this. Fine, let's move on. So these digestive enzymes are actually super powerful and they are capable of breaking down all the organic materials. So whenever there is a disturbance in the cellular metabolism, or we can say when the cell is damaged, these lysosomes burst open, releasing their digestive enzymes and killing their own cell. I know that's sad, and this is what exactly a suicide bomber does, right? And that's why the lysosomes are known as suicide bags. I hope you understood this. You can also think of the lysosomes as the cleaner or the waste disposal system because they help to clean the cell by digesting all the foreign particles like the bacteria, maybe food, and also the old and worn out cell organelles. Well, it seems like the cell is pretty clean now, right? All thanks to the lysosomes. Hey, now out of nowhere, I am gonna ask you a quick question. Who is the powerhouse of the cell? Think. Fine, I'll give you the answer too. What option do I have? So it's the mitochondria, which is known as the powerhouse of the cell. And it's because they are primarily responsible for the synthesis of ATP or adenosine triphosphate molecules, which is the energy currency of the cell. And you might think why it is known as the energy currency of the cell, right? It's because the cell uses the energy stored in the ATP molecules for all the chemical activities and the mechanical works taking place within the cell. I know, it's interesting. And now, if we talk about the structure of the mitochondria, it is double membranous in nature, which means it has two membranous coverings. Here, if you look closely, you will find that the outer covering or the outer membrane is actually porous in nature, and that's why it allows the entry and exit of materials to and from the mitochondria. And now on the other hand, if you look closely at the inner membrane, you can see that it is deeply folded, and these folds actually help in increasing the surface area for the ATP production. Well planned structure, right? Hey, you know what? Mitochondria is the only organelle in an animal cell apart from the nucleus which has its own genetic material, that is DNA or deoxyribonucleic acid. Well, apart from this, they also have ribosomes within them, which are the sites for protein production. So the happy news is that the mitochondria has its own DNA and that it can also create its own proteins. Hey, oops, what is this? I guess it's the plant cell. And what is within this plant cell? Well, this is a vacuole, and as you can see, it's somewhat large, right? It actually occupies 50 to 90% of the entire plant cell. Well, it's quite visible, I suppose. And you know what? I have studied about this vacuole. It is filled with cell sap or a fluid substance that provides rigidity and turgidity to the plant cell. And the vacuole is also known as the storehouse of the cell because it stores amino acids, sugars, and different organic acids. Well, sometimes it stores proteins too. Now, on the other hand, vacuoles are generally smaller in size in the case of an animal cell. Well, if we talk about some other single-celled organisms, like maybe an amoeba, here the vacuole acts as a food vacuole and stores the food consumed by the amoeba. Last but not the least, there are some other unicellular organisms who use the vacuole for removing the extra waste and water from their body. Well, that's a huge lot of work, no? Hey, do you remember plastids, about which we studied earlier when we saw the comparison between the prokaryotes and the eukaryotes? Well, plastids could not be found in an animal cell but could surely be found in a plant cell. And you know what? They are of two different types. The colored plastids are known as chromoplast and the colorless or white ones are known as leucoplasts. Here, chromoplast is actually known as the colored plastid because it contains colored pigments within them. For example, if we talk about a chloroplast maybe, which is a type of the chromoplast, here it contains a green pigment known as chlorophyll. Well, apart from the green pigment, it contains yellow and orange pigments too. But note the point that the green pigment is predominantly found in the chloroplast. So remember that chloroplast is a type of chromoplast that is a colored plastid. Remember that this chloroplast is highly important for the process of photosynthesis because it contains different pigments like chlorophyll. I know that we have studied about this already, but it's just for brushing things up. Okay, so the chloroplast also contains stroma that contains numerous membranes embedded in it where different types of processes takes place. Well, I guess that's all about the chloroplast. Now let's talk about the colorless plastids that are leucoplasts. You know what? They are exclusively responsible for the storage of starch, oils and protein granules. Well, there is one more very important thing that you should definitely remember. That is, plastids also contain their own genetic material just like the mitochondria. And they also contain ribosomes. So obviously they will contain proteins too, right? But remember that plastids are only found in plant cells and not animal cells. Okay? Hey, a quick question. Do you know some more differences between a plant cell and an animal cell? Well, we have already learned about three major differences. That is, the plant cells contains plastids. Now, on the other hand, animal cells do not have plastids. Now, the second point is that plant cells have an extra tough covering, which is known as a cell wall, and this is found above the cell membrane, which gives it extra protection. Now, on the other hand, in an animal cell, this cell wall is absent. Last but not the least, plant cells have bigger vacuole because they are associated with more storage, right? Now, on the other hand, an animal cell has smaller vacuole in comparison to the plant cell. Well, that's all about the three different points. And now let's see some other very important points. Now it's time to learn about some new points. That is, if you look really closely at a plant cell, you will find that it is rectangular shaped or maybe square shaped. And an animal cell is actually round or irregularly shaped, right? So that's one more difference. And now the next difference is that the plant cells rarely contain the lysosomes. But in the case of animal cells, they always have lysosomes within them. So that's the basic differences between these both cells. Okay champs, let's try to conclude what we have learned so far. We saw the cell's ability to function with its organelles and membranes in a specific manner, right? Here we saw that these organelles and membranes actually help the cell to perform functions like respiration, and it also helps in obtaining nutrition, for clearing out waste, and also for making proteins. That's a huge lot of work, right? Well, now you know that why it is known as the fundamental structural unit, because we organisms are made up of these cells, and it is known as the basic functional unit because they perform all the different functions within our body. Okay, so now out of nowhere, I am gonna ask you a question. Do you know how our skin is repaired after getting a deep wound? Well, the answer to this question is cell division. It is the process by which new cells are made from the old cells. That is, the cells divide and form new cells. And you know what? After the cell divides, it actually replaces the old and worn out cells. So this is what happens when we get a wound or a cut. Here new cells are formed. That's interesting, right? Well, this cell division is of two different types. That is mitosis and meiosis. In mitosis, the mother cell will actually divide into two identical or similar daughter cells. And here, note the point that the daughter cells will have the same number of chromosomes like the mother cell. And you know what? This type of division is usually seen during growth and repair of the tissues. Now, the second type of cell division is meiosis, and it takes place in reproductive cells or gametes. You know what? Here, in the case of meiosis, two sets of cell divisions take place, and it gives rise to 4 new daughter cells, unlike in the case of mitosis where only 2 new daughter cells were formed. And there is one more very important point that you should definitely remember. That is, here if the parent cell contains 2n number of chromosomes, then the daughter cells will contain half of it. That is, it will only contain n number of chromosomes, not 2n, okay? Let me ask you a very interesting question. Do you know why the number of chromosomes gets halved in the reproductive cells during meiosis? Well, I'll tell you. It's because in the case of gametes, the sperm and the egg, both carrying n number of chromosomes, will come together during fertilization for forming a zygote that will contain 2n number of chromosomes, which balances everything. So now you know which type of cell division is required when growth and repair of tissue is there, and which type of cell division is required during the formation of gametes or reproductive cells, right? Well, for growth and repair we need mitosis, and for the formation of gametes or reproductive cells we require meiosis. Yay! We got the answer. So with this we have finished the whole chapter, and I hope that you guys had fun time learning this. Well, be good and revise everything, okay? So stay tuned. Bye-bye.